Greetings, Jennifer Doudna. Long time no see, and I hope that you've been doing well during this pandemic time. I'm so glad that you're here with us today to talk about what's happening in your life.
Well, hi, Frances. It's always a pleasure to see you and talk to you. I'm doing very well, thanks for asking. It's been a crazy year for everybody. I would say I'm very grateful for mRNA vaccines and all of the fundamental science that brought us to that point.
Aren't we all? I feel freed now that I'm vaccinated. Of course, we're not free quite yet, but my laboratory has gone back to full capacity. Have you managed to do that as well?
We have not done that yet at Berkeley, but we're maybe within 2 or 3 weeks of opening up a bit more. It's been a bit slower here in the Bay Area. I think there's a real sense of caution, which honestly, as eager as we are to get back to normalcy, I'm grateful for the caution.
Well, I'm completely amazed that in the midst of all this confusion from COVID, that you were able to pivot your whole laboratory over to COVID research. Can you tell us how that's going and what's come out of that?
Well, I have to say that it's been an extraordinary experience for me of teamwork. And this has been a great opportunity for scientists everywhere, really, but certainly in my own lab and my own community here in the Bay Area to step up and use our expertise to address the pandemic. And what made that possible was the Innovative Genomics Institute, which we founded a few years ago in partnership between UC Berkeley, UCSF, and the Gladstone Institutes, with some foundational funding from Mr. Li Ka-shing. And that gave us a team of people who were able to quickly adapt to our changing circumstances a year ago, and among other things, helped us set up a clinical testing lab at UC Berkeley, never done before. So now we have a CLIA-approved clinical lab that handles patient samples. We do all the testing for the Berkeley campus, as well as for several community partners around California, firefighters and other first responders. And we've also been actively working on CRISPR-based diagnostics, new tools to detect viruses, not only the current virus, but of course, preparing for future pandemics.
Oh, that's so exciting. It's a testament to your leadership that you could get people to really move in that direction. What's the status of the diagnostics, and are they becoming commercialized?
Well, the diagnostics are interesting because they build on the natural capability of CRISPR. So what better way to use it than for diagnosing, detecting, and reporting on the presence of a virus? One of the challenges has been to use CRISPR in ways that will quickly detect the presence of the virus RNA and signal, that it's here. And so we're doing this in partnership with it's a 5 academic lab consortium that spans the San Francisco Bay and includes leadership at the Gladstone Institutes with Melanie Ott's team and Dan Fletcher at Berkeley, and then Patrick Hsu, David Savage, and myself at the IGI in Berkeley. And we've been developing 3 different CRISPR-based chemistries as diagnostic tools to allow what we call point-of-care testing. So that means having a little, device sitting on a desk somewhere that allows quick analysis of saliva or nasal swab samples and reports in real time on the presence of the virus. So the strategy ultimately is to provide ongoing surveillance even as everybody gets vaccinated. We feel that for the foreseeable future, we're going to want to know where this virus is for peace of mind.
Well, isn't nature brilliant that she was able to provide these molecular tools, but you're just as brilliant because you know how to use them. I bet everybody in your group wanted to jump in on these projects. How did you apportion that?
Yeah, great question, Frances. It's true that many people were very excited to get going on this. We put up a quick website calling for volunteers, and I think within the first 24 to 48 hours, we had 800 people that wanted to be involved. It was just incredible, very exciting to see that kind of enthusiasm to be working together to face and deal with the pandemic. And since then, we've now been able to turn our clinical lab into a professional lab. We have full-time staff who run the testing operation. But students and postdocs continue to drive the experimental work, of course. They're innovating, figuring out not only how to make CRISPR diagnostics better and make them really work effectively for this particular virus, but also coming up with new ideas. How do we innovate for the future? Because, of course, that's where science, I think, offers the most, doesn't it? It's the creativity, the sort of unexpected idea that develops in a new way that, goes in a direction none of us could predict.
Oh, well, you have a long history of that, and I'll bet the future is going to be very exciting. We're going to talk about that in a bit, but I have to ask you, I've been dying to ask you. I just finished reading The Codebreaker. What's it like to have your life laid bare by Walter Isaacson, along with the CRISPR story?
Well, you probably know me well enough, Frances, to know that I'm not a attention-seeking type of person, and this is a bit disconcerting, honestly, to see my face on the cover of this book. That being said, I guess what attracted me to the project when Walter first contacted me and said, "What would you think if I," and I said, well. What would be exciting to me is if you could really tell the story of discovery in science and talk about the people that were involved in it. Because for me, that's really what makes science such a wonderful adventure. It's an endeavor of people, and it really is all about both collaboration and competition, isn't it? And really, I would say both of those things contribute to the kind of advances that, you and I have both been involved in. So for me, that was what made it a compelling project. And I do feel that Walter did a great job of capturing the energy and the kind of the excitement around both fundamental science, which is what led to CRISPR in the first place as a technology, and then all the things that came after that, including patents and companies and all of the kind of exciting developments that are now happening in the field.
Well, his timing was perfect. In fact, right at the end of the book, you're getting your Nobel Prize. Did he know about it before we all did?
I wonder.
His timing was great, and so the book really ends with this wonderful event, the Nobel Prize, but then ends with the future. So tell us a bit about we have the history of CRISPR. What's the future?
I think the future is a combination of new science, new fundamental discoveries. Some of it will be enabled by CRISPR for sure, but also we're seeing increasing convergence of different kinds of technologies. I'm very excited about the opportunities with machine learning and the ways that data are being managed now. CRISPR is, when it's used in a research mode, is uncovering the functions of many different kinds of genes and pathways that could never have been really effectively probed in the past. However, it does produce enormous amounts of data. And so this is where I think, having other technologies that can mine those data and help us understand what we're learning about the genome, whether it's the human genome or the magnolia genome or anything else, I think that's really important for the future. I also think that increasingly I hope this is true, Frances, that there's just an appreciation for the fact that a lot of fundamental discoveries come from unexpected directions. They come from science that, in our case with CRISPR, with working with Emmanuel Charpentier, and we were just excited to discover the functions of molecules that were part of the CRISPR immune system in bacteria. There was no anticipation early on where that project would lead. And so I think there's a sense that that kind of science will drive future discoveries, the next CRISPR, whatever comes beyond where we are today.
I love how in The Codebreaker you describe the importance of the people who are involved in these discoveries. What was your favorite collaboration of all the many that you've done? Was it the people who drove that and that joy for you, or was it answering the question?
Boy, tough questions. I've, as you said, I've been involved in a number of collaborations over the years, so it's hard to pick my favorite, but I'll talk about one that's very relevant to the story in the Codebreaker, which is our work with Emmanuel Charpentier. And I think the wonderful thing about that collaboration is the way it started. We began as two scientists who met at a conference. We did not know each other ahead of time. We had an idea over lunch in old San Juan, Puerto Rico, about studying the function of a particular protein called CRISPR-Cas9. And that idea and that one conversation led to all of the subsequent work that we did together with our lab members, writing a paper that described CRISPR as a tool for genome editing and how it functioned in a chemical sense. And what I loved about that collaboration, honestly, was really both the fact that the science was incredibly exciting and compelling, but also that the people involved were really interesting people. They were all, of course, very clever, inventive scientists, each one of them, but each of us came from a very different background. It was an international collaboration. Emmanuel is Parisian originally, but was at the time working in Sweden. Her student, Christoph, was working in Vienna, Austria, at a lab there, so we were communicating regularly with him. And in my laboratory, Martin Jinek, who was a postdoctoral scholar at the time at Berkeley in my lab, was from the Czech Republic. One of the many sort of wonderful aspects of that collaboration is that when Martin Jinek and Christoph Chylinski began working together, and Chris in Austria, Martin in Berkeley, they quickly realized that they had grown up on opposite sides of the Polish border, and they each spoke the same dialect of Polish. And so who could have predicted, right? It was just one of those wonderful things. But as you know in your own work, when people hit it off, when there's good chemistry between people, that's often what makes collaboration successful in my experience.
Let's talk about chemistry. You just won the Nobel Prize. I have a feeling I know a little bit about what it's like and what it means, but I'd be curious to know, I know you haven't had it for too long. What has it meant to you?
Receiving a prize like the Nobel Prize is just, for many people, it is a signal and a symbol of the importance of a particular area of science. For those that are in science, it's maybe not that way, but I think for lots of people that are not scientists, it really does signify something special that they wouldn't have otherwise been aware of. And so I've had many people contact me over the last few months. A lot of them are people I had connections to when I was growing up, girls who I went to elementary school with, for example, who are not scientists, but reached out because they saw this news and they felt something very deep about it. And I think especially for women who see other women receiving something like this, it really does say to them that women's work matters. And I think that that's a very important message, especially for younger students that are maybe dreaming, as I once was, about becoming a scientist.
It's one of those remarkable examples of science that goes from the initial discovery, barely 10 years ago, to all these applications that will change the face of medicine, that will affect human lives. Tell us a little bit about that path from discovery to getting it out to helping society.
I've always done very fundamental work in my own research, so I realized early on that for CRISPR, for me personally, to be able to help CRISPR technology be applicable in the clinic especially, but also in other more applied areas such as agriculture, synthetic biology, that we really needed to build an organization that would be able to support teams and support people like me to interact with folks that are working on the more applied sides of those areas of science. Because of the work that teams at the IGI are doing, it's now possible to really envision how we're going to make CRISPR affordable and accessible widely to people that need it for curing genetic diseases such as sickle cell anemia, but many others in the future, as well as thinking about practical ways to help farmers gain access to this technology or to the kinds of agricultural products coming out from CRISPR that will be enabling in the future.
Can you give us an example of how something like this will get out into people's hands?
Well, let's talk a little bit about genetic disease. I think for many people that hear about CRISPR, that's something that immediately jumps to mind is, can this technology cure genetic disease? And I think the answer is clearly yes. And there are already people walking around who have been treated with CRISPR by companies, and they have been shown to be cured of their sickle cell disease trait, as well as other sort of related blood disorders called thalassemias that also have a genetic origin. So this is what CRISPR is terrific at doing, at taking a disease like that has a well-known single gene that's responsible for it, and just tweaking, correcting the code where it needs correction so that the person never has to worry about that disease again. Instead of having to take a drug for the rest of their life to manage a disease, this is actually going to its source and correcting the source code.
What does it take, though, to go from that idea to actually being implemented in the clinic?
Initially, what scientists did was to show that CRISPR could correct disease-causing mutations that cause sickle cell disease in the laboratory in cells that were taken from patients suffering from this disorder. So that demonstrated that the technology can work. And then showing that it could actually be used to correct the mutation in cells that could be replaced in a patient's body. And so that's currently how it works, is that cells are, blood stem cells are taken from a patient, they are gene edited in the lab, and then returned to the patient. But currently this requires bone marrow transplantation to allow those blood stem cells to reimplant in the patient. So as you might imagine, that means that the cost is high. So it's currently a therapy that requires somewhere between $1 and $2 million a patient. And so what we're envisioning in our nonprofit setting is, wouldn't it be great if there were a way to mitigate that cost, to make it affordable to everybody eventually who could benefit from it? How do we do that? How do we think about that? And so I think you have to come at it from a number of different perspectives. One, of course, is the science and the technology. So we're working hard at the IGI right now on ways of delivering CRISPR gene editing molecules directly into the body so that we don't have to use a bone marrow transplant. So that's one strategy. But I think it also involves education. It involves inviting people who are not scientists to work with us, understand a bit about what we're doing, and to engage. And that really requires a whole team as well. And we've been very actively building a group at the IGI that are doing a lot of public outreach and working increasingly with patients to help them see where the science is headed and bring them along with us.
You and Emmanuel are the 6th and 7th women to win the Nobel Prize in chemistry in all of history. It was an historic event, celebrated all over the world. What's it been like as a woman to reach this pinnacle of science? And what were the rocks you had to climb over for this to happen?
Well, honestly, I think the biggest obstacle for me, Frances, is always my own my own psyche. It's my own, doubts or questions about myself. And I think over time, I'm now in my 50s, but I think, over time in my life, I've learned, strategies for dealing with the voices in my head, if you know what I mean. But I think that's probably been the biggest challenge, really, is just, self-doubt. And I think that's true for many people in general, but perhaps more so for some women, at least, because of the way that we're raised. I think that, women have a disproportionate, responsibility often for their families, whether it's for children or for parents. And that often comes at the same time that we're working on our careers and, building up our research programs if we're scientists. And so I think that has always been something on my mind about, not only for me personally and how to, manage all of that, but also just to help enable future generations. And I don't think there's a one-size-fits-all answer to that either. I think it has to be that we build an adaptable community of scientists who are supportive of each other and recognize that, people are they're dealing with different things in their lives at different times, and we just have to have a system that is supportive of scientists to enable all of us to do our best work.
Well, it wasn't just the voices in your own head. I remember that there were some school counselors that weren't so encouraging early on. That can have a huge effect on a young woman.
You're referring to a story that I think is in the Codebreaker about a 10th grade counselor I had in high school who told me that when I was asked, what I wanted to do in the future, I said, well, I want to be a scientist. And he kind of looked at me very skeptically and said, oh, that's not a good career for a girl, And what he didn't know maybe is that I'm a very stubborn person. And when someone tells me not to do something, that's a good incentive to do it. So I became, if anything, even more determined. For me, at least, those kinds of comments, you can take them in two ways. You can take it as, you can allow it to, build up your own negative feelings or voices. Or you can turn it around and say, well, I see that as a challenge, and I'm gonna rise to that challenge and just see if I can, make that work. I really try to view the challenges that we all face along the way in our careers as opportunities, ways to try to be better. We all have failures, experiments that don't work, papers that get rejected, grants that don't get funded, et cetera. But I think, for me, when those things happen, I try to turn it around and say, well, what can I learn from that? How can I do better next time? How can I rise to that challenge? Frances, one of the things that has been so interesting and kind of unexpected for me about CRISPR has been the opportunity to be involved in startup companies, biotech companies that are developing technologies for particular applications, something I had not done, before CRISPR came along for me. And something I've never told you, actually, is that, I think you are probably the most important mentor that I have had with regard to being a company founder. Because when I first got to know you through our meetings at the Packard Foundation, we talked a lot about companies, and you told me a lot about your work with a series of companies that you had been involved with. And I remember being so impressed with your abilities there to manage both a fundamental academic research laboratory, but also to be thinking always about, where is the science going? How can it have a broader, bigger impact? And in many cases, that happens through companies.
Well, I always like to tell my students, it's not useful until someone uses it. And we find that the way to get people to use it is provide it in a form that's usable. And companies do that so well. And who better to drive that than the people, the young people who've worked so hard to make these things happen? So tell me, you've caught up and surpassed anything I ever did now. What are you, up to 7 companies now that you've co-founded? How do you do this?
It often happens organically. It's through people that are working in the laboratory who have an idea and they realize that for this idea to really have an impact, they need to develop it in a context where they can attract investors, they can build a larger team, they can have a focus on a particular idea. And that can be so exciting, as you know. There's something wonderful about seeing young scientists get engaged that way and have an idea they really are passionate about working on. And that's true in academia too. But in these startups that I've been involved in, many of them have been founded by former lab members of mine. And the three in particular that I'm thinking of right now are three that are all led by former students of mine. And that is a real joy to see people who came to your laboratory to learn about doing science and you work together for a few years, and then you see them blossoming and leading a team, raising money, building products and really advancing technology in ways that go far beyond anything they could have done, in my academic lab. I think that is really something that I feel proud of.
Well, some ivory tower scientists might complain a bit over the focus on forming companies. Do you run into that at all?
Not really, no. I haven't really. Now, maybe they're grumbling behind my back, it's possible. But I guess I feel that, when I look over my career, I've now been running my research lab for 27 years. And over that period of time, the vast majority of trainees who came from my laboratory are running academic labs, actually. And many of them are now tenured professors. They're highly successful. I feel, very, very proud of what those folks are doing. But equally, I feel delighted when a student says, gee, I feel a passion to develop this idea in ways that probably make more sense to do in a company than in an academic laboratory. So I really see them as just two different, they're two forks in the path. And I think, as you know, increasingly people can go back and forth between companies. I don't think they're companies and, and academics. I don't think it's sort of a one-way street the way it maybe was when I was a student.
Well, I personally feel that there's really no division between so-called basic research and applied research. These are inextricably intertwined, and one can inform the other in a very powerful way. And I think we're seeing that in spades in your own work.
I agree with that, yeah.
Let's talk a little bit about the ethics of gene editing, specifically human gene editing. I know that you've been a real leader in this area, and that is commendable and remarkable and so important. Can you tell us where you are with this now?
I think first and foremost, we have to encourage transparency about the science. I think we have to be realistic about where it's headed. The field is moving very, very quickly. I mean, to see the advances in genome editing just over the last 8 or 9 years has been extraordinary. And where will we be in another 8 or 9 years? It's hard to even imagine, but I think increasingly it's possible to essentially make any kind of edit to cellular DNA that somebody might like to make. And so that just brings along both incredible opportunities, but also risk and responsibility. So I think those have to go together. And I've kind of learned, I guess, over the last few years that, first of all, that people are actually very interested in this. I didn't have any difficulty recruiting and encouraging people to engage in this topic. So that's been really interesting and gratifying. But also it's extraordinarily important. And one of the events that underscored this, which is discussed in the Codebreaker, was the announcement about human embryo editing that resulted in the birth of twin girls who have edited DNA. And I think that really kind of galvanized the international community to realize that, this is not a fantasy. This is not something off in the distant future. This is actually something happening right now or could happen now. And so we need to just understand it better. We need to understand the implications of it. And rather than saying we're never going to go there, we need to say, if we go there, how do we do it? How do we do it safely, ethically, and in ways that will enhance and not harm humanity?
But the fact that it happened and it surprised the entire world is a reflection of how easy it is to use this technology. Do you really think you can self-regulate? And won't there be many more examples just like what happened with the twins in China?
I think that one thing that's been very interesting after that 2018 event with the CRISPR-edited twins is that my immediate reaction after that announcement was to think, oh my gosh, I wonder if this is the first of many, and we're just, the floodgates are open and we won't be able to turn the tide. That actually hasn't happened. I do think there was a very strong reaction internationally that this is unacceptable. Scientific journals are not going to publish this type of work if it's not performed with appropriate ethical oversight, for example. And the other thing is that on the one hand, yes, it's easy to do. On the other hand, the devil's in the details. And when the details of the editing for these twins was revealed, it was clear that the technology is just not at a point where we can control it in a way that would make editing of human embryos safe, even if we decided it was worth doing.
One thing that I'm dealing with in my new role as co-chair of the President's Council of Advisors on Science and Technology is asking how can we use science and technology to help solve these immense problems that society is facing, and climate change and racial inequity, the pandemic. Do you have hope that science and technology can really deal with these issues, and how are you fostering that?
I do have hope, Frances. And I honestly think that as difficult a year as we've all been through, it's really a triumph for science that we are emerging now, or starting to emerge from this pandemic, with the availability of vaccines, as we discussed previously, and also thinking about the opportunities ahead with all of the advances in science and technology that are happening, it seems, on an incredibly rapid pace. I think the example of the RNA vaccines is simply extraordinary. And it's important for people to realize that that ability to quickly make a brand new kind of vaccine, this had never been done before, right? Using messenger RNA to encode a piece of a virus that would instruct the human body to make mount an immune response to it. That comes from the fact that there was investment over prior decades largely by governments and by the US government in fundamental science, the type that you and I both do. Science around, how to make big RNA molecules, how to deliver them into cells, how to modify them chemically so they don't invoke the wrong kind of immune reaction when they're delivered. All of those things came together at the right time to enable this achievement of science. So for me, and I had nothing to do with it at all. It's just me looking at it as an outsider, but admiring it and feeling hope that that is what science really offers for the future. It offers, as long as we invest in the kind of fundamental work that is going to lay the foundation for future discoveries. We don't know what they are right now, but we know they're out there. And that's what makes it so exciting right now to be a practicing scientist and to be proud of our field, proud of our colleagues, and knowing that great opportunities still lie ahead.
Well, you're a very good example of the optimist. Scientists, most scientists are optimists, right? Otherwise, we wouldn't be out there doing these very difficult experiments without the hope that we're going to uncover some new feat of nature, some new principle of nature, or solve a problem. Well, Jennifer, as usual, it's been an absolute delight to talk with you. I learn so much every time I do, and that's because you're moving so quickly on such important things. So I'm just absorbing your shine. Thank you for doing this.
Well, you're a wonderful interviewer, Frances, and a great friend, so it's been a real pleasure to see you today.